Good afternoon, good evening, and good morning to everyone. I'd like to welcome all participants to this fourth webinar in the series. The title of today's session is COVID-19, Building on the HIV Response to Introduce Innovation in a Pandemic. My name is Lucy Stackpole-Moore, and I'm the Director of HIV Programs and Advocacy with the International AIDS Society, based here in Geneva, Switzerland. Our aim for this webinar series is to provide an opportunity for discussion on the pandemic and have a deeper look at how it impacts people living with HIV, and also connect and celebrate the innovations and the groundbreaking leadership from many IES members, clinicians, frontline health workers, communities, and the greater contribution of science in this evolving pandemic that is changing, as we know, every day in different realities around the world. Before I hand over to the absolutely brilliant set of panelists we have for you and the keynote address for today, I'd just like to run through a couple of housekeeping guidelines for today's session. Please note that due to the high number of attendees, all participants are muted, but do feel free to send your questions through at any time to any of the speakers through the Q&A box at the bottom of your screen. You'll see there's a chat box. That's really if you're having any technical issues in terms of hearing the presentations, seeing slides, or seeing the videos of the speakers. But any technical questions for the Q&A session, the question and answer session, please do post in the Q&A box at the bottom of the screen. You'll notice that there's a plus symbol when someone else has asked a question. And if you have the same question or think it's a really important question, please do click on that plus symbol. That will be a signal to us that that's an important one that we should prioritize in making time to address in the Q&A slot at the end of the webinar. As I said, if you are having any issues hearing or through connecting on Zoom, please do share that information through the chat box at the right of your screen for any technical connection challenges. At the end of the session, we do have 30 minutes for question and answers, and as much as possible, we will get to the questions, or please do keep an eye on the Q&A box, as some of the panelists may be able to respond in writing as well during the webinar. Please do keep the focus of your questions on the focus of today's webinar, which is innovations. And if you have any other questions, we would encourage you to send them via email to coronaquestions@iasociety.org. And also, this is the fourth webinar in a series, and perhaps your questions have been answered in previous sessions or on the COVID-19 portal on the IS website. You'll see the URL there on your screen, so please do check that resource for answers to your questions or other links to really useful aspects of the topic in relation to COVID-19 and HIV, which is available for you to access at any time. I'd also just like to quickly bring your attention to the COVID-19 conference that's coming up and free and accessible to everyone. The submission for abstracts is still open at the moment, and also register, as I said, is free, and please do sign up to participate in that online space in early July. I'll say more about that at the end of the session. But without further ado, I'd like to hand over the moderation to Dr. Raju Tatut, who is the IES Deputy Director in the HIV Programs and Advocacy team, and he'll be moderating today's session. Over to you, Roger.
Thank you very much, Lucy, for this introduction. My name is Roger Tatut. I am the Deputy Director at the International AIDS Society working in the HIV Program and Advocacy Department. I would like to start by thanking all participants who connected from around the world. We had over 1,200 registrations, and currently there are over 360 people who have joined this call, and I am sure more will join. I would also like to thank our presenters for their time and commitment, especially considering the time difference. It is 5 a.m. on the west coast of the US and 10 p.m. in Melbourne, Australia. Webinar on innovation in a pandemic will start with an introductory talk by Professor Sharon Lewin, and will be followed by testimonials from people directly involved in the response to the COVID-19 pandemic. Our speaker and panelists today are Professor Sharon Lewin, Philippe Jacon, Dr. Jim Rooney, Maureen Luba, and Dr. Mookho Malahleha. I'd like to remind all presenters to stay online for the panel discussion and Q&A that will follow the presentation. It is now my pleasure to introduce Professor Sharon Lewin, an infectious disease physician and scientist who has worked for over 25 years in HIV-related clinical medicine and research. Professor Lewin is the director of the Peter Doherty Institute for Infection and Immunity, a joint venture of the University of Melbourne and Royal Melbourne Hospital in Australia. Professor Lewin is also an active clinician working at the Alfred Hospital in Melbourne and a professor of medicine at the University of Melbourne. I'm handing over to you, Sharon.
Thanks very much, Roger, and a great honour to be doing this talk, and congratulations to the IS for their leadership, not only in the HIV response, but now with COVID-19. And the title of my presentation is Innovation in a Pandemic, 40 Years of HIV to the Rescue. And if you could advance the slide, Laurent. So the objectives of the webinar today are to understand the role of innovation in science in the COVID-19 response, to understand where this innovation came from and how was it mobilised so quickly, to highlight what role our experience with HIV played, and whether the COVID-19 response will enhance the HIV response in the future. And I'm going to focus my talk or introductory remarks on these last 2 points, the role of HIV and the impact of COVID-19 on HIV. The next slide, please. So I'd like to first dedicate this talk to a friend and colleague, Professor Geeta Ramji. Geeta was a friend of mine. She was a member of the Conference Organising Committee for AIDS 2014, which I was a local co-chair for in Melbourne, Australia. She was the science co-chair and a wonderful woman. She was previously the Chief Scientific Officer of the Oram Institute in Johannesburg, and unfortunately died of COVID-19 in March 31st of this year. She didn't die at the front line. She died after acquiring COVID-19 while visiting her son in London, and then died on her return to South Africa. A very great loss to the HIV response, of course, a great loss to her friends and family, and a real tragedy that this has happened. Along with Geeta, there have been many other healthcare workers at the very front line, and I also dedicate this talk to them. Next slide, please. So the impact of COVID-19 on every aspect of our lives has been significant. Each of us are sitting in our homes around the world. Our lives have stopped, our work has stopped, our social lives have stopped. And the impact of COVID-19 has affected absolutely every single business, but particularly research and development, and therefore will have a big impact on our response to HIV. I wasn't able to get some really good data from across the world on this level of impact, but some of this modelling has been done in Australia, a relatively small country, of course, of 25 million people. But just in 2020, the estimated loss in Australia just to the university sector, a major contributor to our research and development, is estimated to be $2.5 billion, a loss of about 14,000 jobs, and some serious concerns about the long-term impact on our research workforce. Now, if this is happening across the board in every area of research and development, the impact on our own advances within HIV will also be significant. Next slide, please. The good news story, though, is the impact that HIV has had on COVID-19. And I think over the course of the panel discussion today, you'll hear this loud and clear. It's had a big impact on both the biomedical response to COVID-19, and I hope the non-biomedical or patient-centered response that I hope we will continue to see in our response to the pandemic. So the first, in the areas of biomedical impact, there's no doubt that advances in HIV testing has had a huge impact on COVID-19. Many of you will know that to diagnose COVID-19, you need PCR to detect the virus, and a lot of work being done on antibody tests. Both of these have been absolutely critical in our response to HIV, with significant advances in technologies. Genomics will soon play a very big role in COVID-19 understanding epidemiology. But probably one of the most important advances that HIV has brought is the capacity for near care or point of care and even home-based testing. And we'll be hearing from Philippe Jacon later from Cepheid, and who's been able to translate the advances from HIV testing into COVID-19. Next slide. The other very significant area of biomedical advances from HIV that is currently having a major impact on COVID-19 is in the area of antivirals. Many of you will know that repurposed HIV drugs surprisingly have an activity against COVID-19, specifically HIV protease inhibitors. And while these have not yet shown benefit in randomized clinical trials, potentially in combination with other antivirals, we may see an effect. The whole principle of combination antiviral therapy has been fine-tuned in the management of HIV, and I think will almost certainly be relevant to COVID-19. And then in prevention, PEP and PrEP will also both be important strategies should we not have a vaccine. And we just first of all need to have effective antiviral drugs. And finally, the whole issue of costing of antivirals, again, developed by, in the field of HIV, allowing for a two-tiered system of drugs available in high-income countries and at much lower costs in lower middle-income countries. Some interesting work being developed on costing already for antiviral drugs that will be extremely important once we have an antiviral drug available for COVID-19. And the next slide. And finally, vaccines, probably the hottest and biggest challenge that we need to solve for COVID-19. Many of the delivery vectors that are being tested have been developed for HIV, along with the adjuvants, an enormous amount of thinking in delivery of trial design that will impact in how COVID-19 vaccines roll out. Next slide, please. COVID-19 treatments will likely be far more complicated and a little bit more difficult than what we experience with HIV. In HIV, an antiviral drug rapidly reduced viral load to undetectable levels, allowing immune recovery and return to normal health. It might not be that simple with COVID-19. And this is just a simple schematic showing you the different phases of disease in COVID-19. An initial early stage characterized by viral replication and late stage characterized by inflammation. Making the decision of when to start treatment, what to use, whether to use an antiviral or an anti-inflammatory drug, quite complex, and still this is an area of rapid evolution. Next slide, please. And here is a summary of the COVID-19 vaccine approaches currently in clinical trial. 10 phase 1 and 2 studies distributed around the world using a range of different vaccine approaches. And the list of vaccines that are in preclinical development is huge, over about 100 different vaccine potential strategies. And those that have already reached phase 1 and 2 trials have really been heavily influenced by experience already in the HIV vaccine world, particularly the use of DNA or RNA vaccines, the use of non-replicating vectors like adenovirus, chimp adenovirus, and MVA are all being now tested for COVID-19. So an enormous impact of all that investment that we've made over the last 3 decades in HIV vaccine development. Next slide. But where I hope HIV will have the biggest impact is in the non-biomedical response to a pandemic. And many on this call will know these issues well, but I'd like to remind this audience that the emergency use authorization at the FDA was a result of advocacy from HIV advocates in the early, very painful and dark days of HIV, where there were no treatments available. This led to a new designation for rapid approval or access to drugs prior to approval, and this has already been it's already happened this year for drugs for COVID-19, specifically hydroxychloroquine and remdesivir, and I think this contribution is really, really significant. Next slide. And of course, keeping human rights at the centre of a pandemic response, something that we should never forget and cannot forget in our response to COVID-19. COVID-19, even in high-income countries, is exposing the great differences we have in human rights for different populations. whether it's migrant workers in the very high income country of Singapore, or people of colour in the United States suffering from poor outcomes from COVID-19. We're yet to see that how human rights, or that people that have poor access to treatment or vaccines will suffer from this outbreak. And we need to keep those same principles that have guided the HIV response in the response to COVID-19. Next slide, please. So what are the opportunities for HIV in a post-COVID world? At the moment, our work in research particularly, and even in clinical care, has really stalled in HIV, and that's bringing people a lot of great concern. But there may well be opportunities in a post-COVID world. First of all, there almost certainly will be high global awareness of viruses and the need for pandemic preparedness. No country has been spared from COVID-19. In our response to COVID-19, we will need health systems strengthening globally, and this almost certainly will have benefits in other infections such as HIV. Almost certainly we'll see new global initiatives for testing, drugs and vaccines, and I hope that that scientific innovation COVID-19 will be high, and we are almost certainly likely to see a reciprocal flow back to the HIV sector. Thanks very much.
Thank you, Professor Lewin, for this great introduction, and in particular for raising the issue of long-term losses caused by COVID-19. This is certainly not without reminding us of the early days of the HIV epidemic, although with COVID it's a very different disease. A key point of the response to an infectious disease is our ability to actually detect that undiagnosed that people are being infected. And our next presenter is Philippe Jacon, who is a senior vice president for global access for Cepheid, a company specialized in automated molecular diagnostics. Philippe joined Cepheid in January 2013 and leads the company's strategy to ensure access to Cepheid molecular diagnostics globally, with an emphasis in low and middle income countries. Prior to Cepheid, Philippe holds the position of CEO of the Foundation for Innovative New Diagnostics, also known as FIND, since October 2011. He joined FIND from the European Diagnostics Manufacturer Association, where he acted as the Director General ad interim since March 2011. Philippe has a microbiologist engineer degree from the Université de Bretagne Occidentale in France, and I'm handing over to you, Philippe.
Thank you, Roger. And again, I want to thank you really because of your willingness to have me on the panel representing the diagnostic companies and the diagnostic industry. I think the COVID crisis has opened the eyes of many about the critical importance of diagnostics in the management of such a crisis. And hopefully this eye-opening is going to remain when it relates to all infectious diseases. So I'm going to start by giving some basic information building on what Sharon presented earlier. And as we know right now, the pandemic is still in active mode, and it's important to test symptomatic patients. And when we found positive cases, then test the people who have recently been in contact with these people. And in order to do that, the tests used are molecular tests. These tests are run on the same type of equipment that is running HIV tests currently. They can be either manual technologies requiring specific lab infrastructure, well-trained technicians. They can be more automated using high throughput, well-trained technicians, also batch mode type equipments. But they can also be more decentralized with technologies like our GeneXpert that can be also run by less specialized technicians. But now, molecular tests are not the only ones, as Sharon was explaining before. So there are other types of tests. First of all are the antibody testing that will indicate if an individual has been in contact with the virus and therefore has developed antibodies against that virus. And, most likely, as it happens with many viruses, and not yet proven, I believe, but showing some kind of protection of that individual, at least for some time. Finally, there are also antigen tests that are in development. They are not yet endorsed by WHO, for instance. Their principle is also to look at active infection, but clearly it takes a bit more time to detect a positive, and the sensitivity of the test is usually lower than the molecular testing. So, it's good to have all these tests, but just to get back a little bit on how did we get there. The COVID tests have been really developed in a record time. On average, it takes about 18 months to develop a test from the beginning up to registration and being able to sell the test in countries. In the case of COVID, the tests have been developed in less than 2 months. This is absolutely unbelievable, and including our test. And from the beginning to receiving the emergency use authorization from the FDA. So how can we do that? And I cannot explain exactly all the details of all the tests, but I know that as far as we're concerned, we used the same architecture for the COVID test as the test that we have for flu RSV, and that helped us really cut time to get to the product finalization. Something I really want to emphasize on molecular testing is that the test for COVID is really run today on the same machines that are running the HIV testing. So this is very important to note, because certainly there is some leverage that can happen right now in this way, meaning using the machines that are used for HIV, but probably because countries are going to procure additional machines that later on, when the crisis is over, the same machines can be used to test more for HIV. So I think this is important to understand the polyvalency of these platforms and the fact that they can be used for different types of infections. Certainly, as far as we are concerned, we have a very large installed base of systems across the world in developed and developing countries that have been rolled out mainly to support our HIV markets, but also the TB market. And all these machines can test for COVID. But all that said, I think developing a test is not the end of the story. When you have a test, then the question is, how will these tests get onto the market? And also to make sure that people that are going to use the test are properly trained to know how to do it. But first of all, I want to make sure that we say it, because it's one of the challenges that everybody is facing today. The whole industry, the whole diagnostic industry, is faced with limitation of capacity, given that all countries around the world are asking for the COVID test. And this is a bit difficult for all of us to ensure that everybody can get the number of tests that they require. So we are all working with the so-called diagnostic consortium under WHO, which is also looking at the allocation of the tests around the world to make sure that countries get their fair share of the products manufactured. But even with that, and this organization that has been put in place, there are other challenges that need to be addressed. First and foremost is certainly the challenge of logistics. Even the large procurement agencies have still difficulties today to find flights or cargo space to move products to certain countries. It's very difficult. The second issue or challenge that we still see in some countries, hopefully not too many, but is the regulatory barriers, that some countries do not change because of COVID, so it takes a long time to get registration, or they agree to get some kind of waivers for a short time, but it takes very long time to get these waivers in place. So this is something that we need also to keep in mind. There is not just the test, but many different things that we need to do to make sure the tests get there. What also is of key importance is to keep that concept in mind, that there is no good diagnostic test without a good sample. and the good sample is taken using swabs. The most typical one is the nasopharyngeal swab, which is this very long Q-tip type swab. And there is also a shortage of these nasopharyngeal swabs around the world, which is making things even more difficult. So I have to say that we're very pleased at Cepheid that we've been able to get authorization for a nasal swab, and also, to use a saline as a medium to transport the virus, as opposed to the viral transport media, which is the preferred one usually. So all of that are a set of challenges that needs to be put, or to be understood, and we need to find ways to address them before we can make sure that the diagnostic tests are really reaching out to all the people around the globe. So with that said, I'll be happy later on to take any questions during the Q&A session, and I thank you for your attention.
Thank you very much, Philippe, for this presentation. As you noted, testing is not just about developing a tool for diagnosis, but also about providing a lot of things around this tool, from training of technicians and making sure we can ship reagent and machine. And our next presenter is Dr. Jim Rooney, Vice President of Medical Affairs at Gilead Science. Dr. Rooney joined Gilead Science in 1996 as a Vice President of Clinical Research, and in 2004 became Vice President of Medical Affairs. Dr. Rooney has been instrumental in the clinical development of the company antiviral therapy, including treatment for HIV, hepatitis B, and influenza. Dr. Rooney also leads many of the company's clinical research activity in the developing world. And Jim has received his medical degree and residency training in internal medicine at the University of Maryland in Baltimore, and his training in infectious disease at the Laboratory of Clinical Investigation at the NIH in Bethesda. Jim is also board certified in internal medicine and infectious disease, and a fellow of the Infectious Disease Society of America, and will talk to us about innovation in developing drugs. Over to you, Jim.
Jim, sorry, I think you're on mute at the moment. It's difficult to hear anything, but we can see you clearly.
So sorry. Great. My Zoom skills should be much better.
Welcome back.
After all this experience over the past few weeks, but thank you very much. Hi, everybody, and a pleasure to be with you here today and to discuss our response to this terrible pandemic, and what we've learned from our experience with HIV and how that's informed our ability to move forward more rapidly in responding to this incredible and unfortunate pandemic. In our response in HIV over the and other viruses over the past 30 years or so, I think has been key in terms of our ability to learn how to more efficiently, more quickly, and more effectively move forward against this terrible pathogen. I think the ability of industry, scientists, academia to develop over 30 medications now available to treat HIV has been a great experience, and we've learned a tremendous amount from that. That's allowed us to move forward rapidly in response to the SARS-2 coronavirus. Several key learnings, I think, that we've had over the past 30 years have been a better understanding of viral pathogenesis and how viruses cause disease, how they interact with the human host, how they take over our host cell machinery to produce more viruses and induce the pathology that they do. And in this understanding of viral pathogenesis, it allows us the ability to identify potential targets for therapeutic intervention. And advances in science over the past 30 years have allowed for rapid identification, for example, of the viral sequence. We learned within weeks of the time that the virus has identified what the sequence of the virus was. And it allowed us a tremendous amount of insight into potential approaches to therapeutic interventions. Once we knew the sequence of the virus, we could identify, based upon past knowledge of similar coronaviruses and of pathogenic coronaviruses like SARS and MERS, we could identify fairly quickly targets that we thought could be important targets for intervention. And those would include, for example, the enzymes involved in virus replication, such as the viral polymerase. In the case of SARS-CoV-2, it's an RNA-dependent RNA polymerase, and SARS-CoV-2 also has proteases. And we learned from our experience with HIV that two of the most important targets for therapeutic interventions were the enzymes involved in virus replication, and in the case of HIV, the HIV reverse transcriptase, and in the case of proteases, the HIV proteases. And they were targets that led to some of the first and most important interventions that allowed us to gain control of the virus, reduce virus replication to levels of undetectable, and significantly reduce both morbidity and mortality. So literally within weeks of the time of identifying the virus, it was possible to get a view, to get a vision for what might be potential therapies going forward. So that I think is reflective of the advances in our ability to understand both viral pathogenesis and the tools and technology to do rapid sequencing, and also to our experience in terms of identifying potential targets for intervention. In addition, there are several other key elements that that have allowed us to move forward rapidly. For example, our companies oftentimes will have available a knowledge of and have a library of compounds available that potentially could be used to address or to intervene against these potential targets. And knowledge of structural chemistry has advanced substantially in the last 30 years, so that one could identify, when one identified a target such as polymerase or protease, the structural components, and even visualize what that molecule would look like, identify both sites of binding pockets and also potentially catalytic sites for the enzymes. And past experience in terms of developing therapeutic interventions, for example, in HIV, allowed an ability to envision what a therapeutic intervention for SARS-CoV might look like. The availability of extensive libraries of molecules allowed the ability to be able to rapidly screen potential therapeutic interventions against these targets. And the ability of new techniques developed, such as high throughput screening, allow the identification and selection from thousands of molecules of a handful that might have the most promising, the greatest ability to bind to these targets and potentially interfere with their normal catalytic or pathogenic properties. In addition to that, companies had models available, both in vitro and also animal models for many viruses, and including the coronaviruses, that would allow them to test some of these compounds in some of the systems to identify those that might have potential promise, and to be able to not only look at the structural and molecular features of these compounds, their binding capacities, their abilities to inhibit catalytic activities, but also their ability to inhibit virus replication either in vivo or in animal models. And that can provide useful information for developing both therapeutic and potentially preventative therapies as well. In addition, our ability to collaborate with each other, academicians, scientists, government researchers, industry representatives, and the community coming together to discuss potential strategies for moving clinical trials forward, to exchange information, ideas, compounds, and other things, to look our experience in HIV also with combination therapies, I think has been key in terms of our ability to envision potential strategies for moving forward with therapeutic combinations that could have more potent antiviral effects and or combine antiviral effects with other types of therapeutic intervention. In particular, in the case of SARS-CoV-2, it appears that there's two key components to the pathogenesis. One certainly is the viral replication and damage caused by the virus directly, but also the host immune response to the virus, which can induce, in some individuals, for reasons that are not completely understood, an overwhelming response that can result in serious pathogenic consequences, such as acute respiratory distress syndrome, coagulopathies, etc., which may be important contributors to morbidity and mortality. So combining not only drugs to improve an antiviral effect, but also a better understanding of the pathogenesis of the disease, to combine both antiviral compounds with perhaps compounds that might modulate the host response to result in a decrease in morbidity and an improvement in mortality. I think those are some of the key elements that have been involved in the response so far. I work for one company, Gilead, that has one compound that has appeared to be hopefully promising. At least in preliminary studies, it appears to have some benefit in terms of reducing the time for hospitalization, and further studies could potentially elucidate the benefit and the timing for both the patient population and also the time for intervention in which it could be most useful, not only from HIV, where we've learned many things about the ability of drugs to interfere with viral pathogenesis, but also from our experience with other viruses, such as influenza. And Gilead was also involved in the development of a drug for influenza, oseltamivir or Tamiflu. We recognize that not only is it important who you select to be treated, but the timing of that intervention. We know that virus replication occurs in acute respiratory infections early on during the course of disease. It appears for SARS-CoV-2, it can occur relatively early on, even before symptoms appear, and you can get relatively high virus titers. So perhaps intervening early could provide the greatest opportunity for antiviral compound to have benefit. And so I think as studies go forward, we'll be learning more not only about the patient population that could benefit the most from these types of drugs, but also the timing of those interventions along with other ones that could affect other components of the disease, such as the immunopathogenesis that's important here. So
Jim, I have to stop you as we are running out of time.
Yes, thank you.
Running slightly over your time allocation. Thank you very much for this contribution. It's remarkable how a work done on one virus has been facilitating the research and the understanding of another virus and developing drug faster. Drugs, of course, needs to be trialed, and it is now a nice way for me to hand over to our next presenter, Dr. Mookho Malahleha. Mookho is a co-principal investigator for the HVTN 702 HIV vaccine studies and deputy director for the Setshaba Research Center in Pretoria, South Africa. Dr. Malahleha is a qualified medical doctor with a master in public health degree and a diploma in HIV management. She has over 10 years of experience in HIV prevention and TB clinical research. She is a member of the Advancing Care and Treatment for TB HIV Consortium, RTI Steering Committee of Multipurpose Technology, and TB Alliance Stakeholder Association. She has been awarded the HIV Vaccine Trial Network Mentorship Award in 2018 in recognition of her effort to train and mentor research staff on HIV vaccine research. Mookho, I hand over to you.
Thank you, Roger, for that wonderful introduction. It is indeed a pleasure to be part of this panel discussion. I will be providing insights on how COVID-19 has affected HIV prevention research in South Africa, and also try and unpack how our approaches to HIV research have laid the foundation for COVID-19 approaches. But before I get into that, I just wanted to provide a country level background in terms of the most recent COVID-19 statistics. And as of yesterday, South Africa had screened a total of 9 million people and had conducted 370,000 tests, which is equally distributed between the private and the public sector. We have 12,000, just about 12,000 confirmed COVID cases in our country, and this is a positivity rate of 2.8%. We have 219 reported deaths, and thus we have a mortality rate of 1.8%. Now, in terms of lockdown restrictions in our country, on the 26th of March, our president imposed a level 5 lockdown, which is the highest restrictive level, only permitting essential services. And we had this level 5 lockdown for a period of 5 weeks. We are currently at level 4, and hopefully at the end of May, we will move to a level 3 of lockdown, and thus more sectors, more economic sectors would resume activity. The one point to make is that the president's risk-adjusted lockdown regulations have been informed by the science, and we need to commend him for this. Now, how has COVID-19 impacted clinical trials, especially HIV clinical trials in our country? So we've had major challenges, especially during the level 5 lockdowns, to conduct study visits. We had challenges with logistics and transportation of trial participants, and also challenges with them accessing other services at local health centers. We were fortunate, however, that our South African Health Products Regulatory Agency, the South African Clinical Trials Regulator, issued guidance in March on how to conduct trials, clinical trials, during this period of COVID-19. And their emphasis on us as researchers and sponsors to put participant safety first, and also to ensure that trial staff were protected and were given essential PPE to conduct trial visits. We're also provided with guidance on what essential visits would be, or critical study visits would be. Likewise, also the ethics committees were engaged as well, and they did also provide similar guidance to the trial sites. Thus, our sites had to explore and adapt our site operations to conduct these visits. Our site staff managed to rotate in groups. They were provided with PPE, despite quite a number of challenges to actually get PPE into our country. But I think ultimately we managed, and most of our trial sites were provided with PPE to conduct visits. The trial centers as well implemented various health and safety protocols to ensure that participant visits were conducted in optimal settings. Within our HIV vaccine trials, vaccinations and post-vaccination visits occurred, and these were prioritized. We also adopted remote healthcare and communication models for visits where possible, and this was via telephonic calls or WhatsApp calling. And this also led us to have further discussions to basically deliberate and ask ourselves whether there's a place for telemedicine, maybe at a small scale in clinical trials, without losing the patient or the participant-centered approach, which has been the success of clinical trials. Other challenges included getting participants to trial sites. And this, I think the sites were tremendous in this, assisting participants to come through and offering transport to get participants to site. So I'll also just touch briefly on how our existing approaches to HIV research have laid the foundation to COVID-19 approaches. If there is one thing that we have learned from dealing with the HIV epidemic, is the need for collaborative approaches. Our clinical trial sites have been at the heart of communities that have been affected by HIV/AIDS, and they have managed to create an imprint or create a footprint in these communities, working with different sectors, I think as Jim has alluded to, working with different ethics committees, with the regulators, with communities, civil society, and also working with the general communities to ensure that we have a proper infrastructure to conduct research. Similarly, this collaborative approach is going to be quite critical in the approaches to COVID-19, and also to introduce various COVID-19 treatment and prevention modalities into our communities. Additionally, in South Africa, we have an extensive community screening and testing program at the moment, which has resulted in 100,000 people being tested for COVID-19. This approach is going to change somewhat from a general community screening and testing approach to targeting hotspots and those at high risk using surveillance data and geospatial mapping. I think those in HIV research arena will appreciate that these are efforts that have been long-standing in HIV research in terms of targeting hotspots and those at high risk. Similarly, social and behavioral science in response to the HIV epidemic has been very critical, and thus we need similar approaches towards COVID-19 to understand the social and the cultural context of COVID-19 disease, to understand risk perceptions, and also to guide on health communication and on acceptability of interventions being researched for COVID. Lastly, community engagement, which is a very important piece of the research puzzle, and it holds a critical place in the success of HIV trials. We know as researchers that we do not work in silos. We need our communities, we need civil society, and thus it will be imperative to engage advocacy and civil society groups in the process of COVID-19 research. I will stop there and we'll be able to take questions later on. Thank you.
Thank you very much, Mookho, for this presentation. It's remarkable to hear about the capacity of sites to adapt and how you responded to the challenge of conducting trials under lockdown. Your final point on the community is very timely. Our final speaker today is Maureen Luba. We are slightly running behind times, but I'm sure Maureen can stick to time. Maureen is the Africa Region Advocacy Advisor at the AIDS Vaccine Advocacy Coalition, better known as AVAC. She helps coordinate civil society organization capacity building effort in Zimbabwe, Tanzania, and Malawi to enable them to effectively engage in global fund, PEPFAR, and national policy decision-making process. She graduated from University of Malawi with a bachelor's degree in public administration. She has more than 10 years of experience working on HIV treatment and prevention and sexual and reproductive health and rights program. There's many other things I could say about Maureen, but for the sake of time, I'm going to stop here, just adding that she's a board member of the International Partnership for Microbicide, and she's also a board member of the Global HIV Vaccine Enterprise Advisory Group. Handing over to you, Maureen.
Thank you so much, Loje. Can you hear me clearly?
Yes, we can.
Okay, thank you so much. So thanks once again for that kind introduction. I'll try to keep time, as you have said. But I just wanted to start by saying that communities were actually the first responders to HIV almost 4 decades ago, and they have continued to advocate for a robust response to the HIV epidemic. And we know that communities have also taken part in the delivery of HIV services, specifically making sure that geographical location, social and economic status has access to those services. So when it comes to HIV research, communities have actually contributed to the discovery of the HIV diagnostics, preventive and therapeutic tools that we have seen today. And they've done that by contributing to the body of knowledge that is
Maureen, we have lost sound with you.
To address the myths and misinformation about therapeutic and vaccine trials. And they've done that by using their knowledge about the science, but also the skills to effectively communicate that information to the broader communities in a language that is so easy to understand. COVID-19, we've seen a lot of this information around vaccines resurfacing on social media and on so many platforms.
As apologies for the break in the sound. I am not even sure that Maureen can hear me. Maureen, let me send her.
Researchers, regulators, and funders, sometimes even way ahead trial completion to ensure that the right systems and mechanisms are put in place to allow for accelerated product introduction once proven safe and effective. And they can even continue to do that the same even with COVID-19 research. So the list is just endless. Now coming to COVID-19 research, as the search for diagnostics, therapeutics, and vaccines continues at such an enormous speed, it's important for us to make sure that the role of communities is clearly defined, but also adequately addressed. We've learned from the HIV field that inclusion of communities in research does not only lead to better science, but it can also facilitate rapid translation of research findings into accepted and effective programs in the real world. But however, for that community participation to happen, the following actually needs to happen. First, having a structured mechanism for consultation and community engagement will be very, very critical. In the HIV research field, such structures have taken the form of community advisory board, and this is something that the COVID-19 research field could learn from. At the moment, TAG and AVAC are working with global partners to develop a global community advisory mechanism that could work with WHO, vaccine researchers and developers to ensure that COVID-19 vaccine research happens to the highest possible scientific and ethical standards. And in addition to that, constant structure and systematic sharing of information with communities on the progress of the different research programs will help to keep the momentum, maintain the interest, but also keep the communities and other stakeholders engaged. What we have seen with COVID-19 is that almost every day you wake up to new information, whether about a new clinical trial or a new discovery. And sometimes it's just so hard to keep track of what's going on in this world. I think we all know that we're dealing with a novel pandemic here, and that more information about COVID-19, including its pathobiology, the immune response. It's important that we continue to invest in the education and capacity building, specifically of communities, so that they can effectively contribute to the search towards effective therapeutics and vaccine candidates, as well as their involvement in the general COVID-19 response. As the different forms are being applied in the COVID-19 vaccine treatment and treatment trials, it is important that the researchers spend enough time to explain the details about how do those therapeutic and vaccine candidates currently in the pipeline work. What is the clinical trial process, right? What are the research ethics and how are they being adhered to? I think one of the things that we have also learned from the HIV field is that maintaining transparency and hope, even in the face of disappointing trial results, has been one of the challenges which the HIV field has grappled with. We all know that not all the therapeutic and vaccine candidates currently in the pipeline will yield to efficacy. So what can we learn from the HIV research field to make sure that such mass messaging is packaged correctly? In conclusion, I just wanted to emphasize that we know the field is rushing to find safe and effective therapeutics and vaccines, but let us make sure that proper ethical guidelines and requirements are being adhered to. I think it's also important that the research field continues to apply and integrate the gender range, but also other relevant community vulnerabilities in the therapeutic and vaccine programs to ensure that the specific needs of different populations, especially those that are affected, are being addressed in the process. Lastly but not least, communities need to be treated as equal partners in this endeavor to find effective therapeutics and vaccines, and not merely seen as potential volunteers for the trials. Thank you so much. I hope I was audible enough.
Thank you very much, Maureen. We had a few breaks during your speech and your talk, and apologize to everybody about that. You may have to put your video off whilst we are having the panel discussion if it improves the communication. Thank you for emphasizing the importance and the role of the community in the response. We're now going to move into the next part of our webinar today, which is a panel discussion, which I will keep a little bit shorter, trying to to keep us to time and give time for Q&A from the audience. We have a number of questions that have arrived in the Q&A box. This is where you should put your question. I think it is clear that the world was taken by surprise by this epidemic, even if a number of people had been warning that this could happen. I think the preparedness is a big issue. Amazingly, as we have heard, thanks to years of research, pre-existing research, development of new tools, we heard about genomics, about structural chemistry, and it seems the innovators were more ready. Nevertheless, there are still some issues and complexity, and I think I'm going to throw a first question to the panel, to our panelists, which is about, it seems the innovators were prepared. How prepared were the regulator, for example? Was that a major issue? And I'm going to ask Philippe if you want to comment on that. I think you brushed the issue, and I'm going to ask all of you to keep your response very short, and we'll move on to other question.
Yeah, and that's a very good question, Roger, and I thank you for asking. I think the answer is a little bit at different levels. I would say that the FDA has a process which is in place, very well understood, of emergency use authorization, that I think most of the diagnostic test manufacturers have been using to get the product on the market quickly. This is not the case everywhere. And on top of that, we are still faced with some regulatory hurdles in country that we should never forget about, as I mentioned earlier. So I think we see now that things might improve. People understand the need to open up quicker, but I think this is a long way from where we are today.
Thank you. Carrying on these topics, innovation may occur within companies, but often university and academics are involved in the process of designing tests or new drugs. And that's a question maybe for Maureen, but also Jim. How much collaboration, to your knowledge, has there been between these two universes, between the academia and the industry, towards developing innovative testing, drugs, clinical trial protocol? Shall I start by Sharon?
I think that varies around the world, of course, about that level of interaction. My experience is that in the academic, and I think in the commercial world there's been an incredible amount of openness and exchange of information. That's been pretty spectacular, I think, in COVID-19. The ability to, the requirement for print publications, open sharing, rapid dissemination of information has been pretty extraordinary.
Thank you. Jim, would you like to add something on collaboration with the university, academia world?
Sure, Roger. No, I agree with Sharon. It's really been quite amazing. The collaboration around coronaviruses obviously extends back to work on SARS in the early 2000s and also MERS in the last 10 years or so. And there were affiliations in place with government, researchers, academia, and industry to help identify both promising therapeutic interventions as well as mechanisms for evaluating those. And I think those were put into place quite quickly. There were also compassionate use programs that were put into place. For example, the drug I mentioned earlier, remdesivir, had a compassionate use program in place for Ebola. It was studied for Ebola, wasn't as effective as some of the other therapies, monoclonal antibodies and others, but nonetheless, there was both a stockpile of drug and also a compassionate use program, so that when SARS-CoV-2 came along and there was some hope that it might also have effect there, we were able to leverage that system to use the emergency, the compassionate use program in the US and the available stockpile to make the drug available quite rapidly to patients. So I think all of the experience that we've gained over the last many years and collaborations and with regulators also has been quite extraordinary.
Thank you. Obviously, I mean, that was discussed during the presentation, introducing this innovation to community. I'm going back to Maureen. Hopefully, we can hear her well this time. What do you think, Maureen, the community can contribute to the introduction of this innovation into community? If we think about testing, for example, what contribution can you make that would actually, and how can we maximize this contribution of communities to ensure that innovation is introduced fairly and delivers quickly?
Sorry, Roger, if you can just repeat that. Very bad connection, so I couldn't hear you.
I was just saying, from your perspective, how can we maximize the contribution of community to the introduction of innovative tools like testing or new drugs, or bringing community to participate in trial? In a very short response, what's the most important things from you? I don't know if you can hear me.
Yeah, so I couldn't hear the whole question, but I think I can still come in. So I think the only way we can maximize, I think, the involvement of communities is to make sure that they're part of the decision making. I think we have seen, even at the global level, there are so many platforms, that are being developed. But where is the community in there, in those spaces? For instance, just to give an example, the ACT accelerator, and there are so many platforms, right? So I think it's important to make sure that I think the different platforms that are being formulated, both at global level as well as at country level, communities should be there. Communities should contribute to the discussion, to the decisions that are being made. Because if you're leaving the communities behind, then we won't be able to leverage on the innovation, the information, the insights that we have, about the communities in which they reside. So I don't know if I addressed, because I only managed to get the first part of the question.
Thank you. I think that's a great response, and it shows that innovation in communication still has some work to do. I'd like to move on about clinical trial. Clearly, COVID is here to stay. Are we going to have to design innovative trial within the context of COVID? How do you see COVID impacting on the future design of clinical trial? Yeah, back with you. Did you hear my question?
Can you repeat that?
Yes. How do you see, because COVID is here to stay, we'll have to probably be innovative in the design of future trial. how do you see the design of the future trial of prevention being affected by the COVID being endemic?
Okay, thanks for that question, Roger. So it's going to be a difficult one, but I think trial sites will need to develop approaches to seeing participants in smaller numbers. It's going to be quite challenging for large-scale efficacy trials where we see quite a large number of participants on a daily basis. But we can develop approaches that we have used currently now to try and attend to participants. And some of these approaches involve seeing participants at different time schedules and additionally implementing various health infection protocols within the trial centers to ensure also that we have social distancing as well. There's a number of different measures and approaches that we will have to look at going forward, but definitely it's not going to be business usual. There's a lot of thought that will have to go into redesigning how we conduct our operations in terms of clinical trials.
Thank you very much. I'd like to give an opportunity to the panelists to, if they have asked questions to one another, or if they would like to respond to some of the comments that were made during the presentation. If you want to raise your hand, or if you want to comment further or ask a question to one another, please go ahead. Sharon.
Oh, thanks very much, Roger. I was going to ask Philippe about the sort of double-edged sword of rapid approval of tests, or what I think has been quite unique in COVID-19 has been this flood of point-of-care tests with variable validation, and in different countries, governments sort of rapidly purchasing these tests. It's happening everywhere in Australia and the UK. It was an interesting phenomenon in the event of an emergency, and all of these manufacturers coming in very suddenly and governments purchasing very quickly. I'd just be interested in your take on it, Philippe. Have you seen something like this before? The way that these point-of-care antibody tests flooded the market and may, in the end, not have a huge clinical utility? Obviously different to the Cepheid test. I'm talking about the antibody test.
Yeah, thank you for the question. Yeah, no, I think we actually have never seen such a response before. And certainly COVID has unlocked a number of ways of responding to a pandemic that nobody could have thought of before. That said, I think we have to be careful. And I'm representing industry, but I want also to make sure that the products that are out there are of good quality. And we unfortunately have seen even in Europe, some of the countries buying some tests that had been quickly developed and having to stop it because of the low quality of the test. So I think we always have to be very careful about making sure that whatever if this is a test or even a drug or a vaccine or whatever, there is a number of things that needs to be validated before we put these tests out there. So I think, yes, to your point, never seen that before. But there is a but, right? We need to make sure that these tests are of a good quality.
Thank you, Philippe. Actually, that was one of the questions in the Q&A. How do we ensure quality of the test in the context of an emergency? So I think that question has been partially answered.
Yeah, and then there are, Roger, there are some bodies that are also validating the test. I think, Sharon, you were mentioning the pre-qualification by WHO, which is one of those. FIND is probably one of the most important players in the validation of new tests like that. But frankly, and we know that because our test is going through it right now, it takes time. And it takes time because there is a long line. And then so they have to do it and find the samples and test according to certain protocols. And so all of that takes time. So certainly when you look at the regulatory question we had before, one of the reasons why the FDA has been able to do that so quickly is also because of the resources that the FDA can put in front of such a pandemic that not many regulatory bodies around the world can do. So again, that's kudos to the FDA, probably lessons to be learned for others. For instance, in Europe, the IVD directive doesn't have any ways to get an emergency use authorization. That said, the IVD regulation that is going to put in place in a couple of years has an emergency use authorization for tests. So I think things are moving in the right direction for sure.
Thank you. Is there other question from one panelist to another? Don't forget to wave your hand.
Roger, I'd like to answer one of the questions that was put up by one of the attendees, and that was, how do we ensure fair and equitable distribution of potential interventions across the globe? And I think HIV has provided a good model for that. You know, many of the treatments that have been developed for both treatment and also prevention of HIV have been available through a variety of novel mechanisms, including generic licensing, as well as contributions of intellectual property to the medicines patent pool. And I would envision that some of those same mechanisms might eventually be used in terms of making therapeutics for COVID available globally. As an example of that, I know the company I mentioned, Gilead, the company I work for, announced just the other day that they had signed voluntary licensing agreements with a variety of generic companies to make remdesivir available across the globe, should it turn out to be safe and effective eventually in clinical trials. So that's one example, but I think the model thing we've learned from HIV can be leveraged here to try to make resources available across the globe and access in a fair and equitable fashion.
Thank you. And I think this question of access came a number of times for a number of products, a number of companies, and a number of technology. Thinking about technology, one of the suggestions we had from one of the member of the audience was lesson learned from HIV testing applicable to COVID testing, and that person was suggesting, you know, why not doing COVID testing in different contexts, such as at the dentist, for example. I don't know if someone want to comment on that, but I think if it would be good to have a comment on how do you implement this innovation in contexts that are different from a medical context. Someone want to say something on that?
Yeah, just a quick comment, because it's about testing, right? I think we also live in an environment where which is very highly regulated. So it's not everywhere that you can run tests outside of a lab and under the supervision of a biologist or somebody who is going to sign the results. And I think this COVID crisis might change a little bit the way this is looked at. We start to see it also for hepatitis, for instance, hepatitis C, where now that there are drugs available, the ability to test for key populations where people are, because these people are out of the normal healthcare systems, right? And it's not by waiting for them to get to a lab that we will solve these solutions, but we need to get out there to find the people. And I think we see now these kind of approaches being more and more tried and accepted in different countries. And I think this is probably one of the ways, and maybe dentist is a good example of what can be done in the future, because people go there, right? And we need to find the people. We cannot just wait for the people to come to the healthcare system.
Thank you. This actually bring to mind something a bit different, but related to acceptability, and that's a question for Mookho. Stigma. So we know there is a lot of stigma around HIV still. Have you experienced stigma around COVID? Have you seen that and how it may have impacted work you may be doing around COVID in trial?
Okay, so Roger, in terms of our country, we have years of experience dealing with stigma, particularly in stigma related to HIV. And I think this has been an important lesson also in terms of approaching COVID-19. We were quite fortunate in our country that early on, our Department of Health and the government coined many different messages for our communities. And these messages were clear messages talking about COVID-19, the transmission, and they were even coined at a level for younger children. And this has led to a whole better understanding in our communities. And thus we see less stigma related to COVID. I think a good understanding and proper communication is key to reduce stigma.
Thank you. You mentioned stigma, education, information. We are living in a time where there's a lot of misinformation around a lot of things, and COVID is, not exempt to that. One of the questions we had on the Q&A, how to deal with the lot of fake news and fear that these generate? And I don't know who wants to answer that. Maybe Maureen, if we can hear you again. How do we deal with fake news, the fear they generate, and the impact they have? I'm not sure Maureen hears me.
Hello?
Yeah, can you hear me, Maureen?
Yes, I heard part of that. I guess, can I come in?
Yes, come in. It's about how do we deal with fake news and the negative impact on implementing innovation or conducting trial.
Yeah, so I think I mentioned that it's very important that I think we need to make sure, I think like I said, we all know that we're dealing with, with a virus that is new, and there's still a lot that we need to learn about this virus. And I think it's important, I think I already highlighted this, that the communities have a role to play in also helping addressing this, the rumors and the misinformation that has been making rounds on social media and so many platforms. And the way we can do that is by making sure that the communities themselves, the advocates, the activists that have led, that have played a critical role, over the years in some, it's not just HIV, but a lot of more, a number of epidemics. We need to make sure that the activists, the civil society, they have the right information, right? And that's why I talked about structured, systematic, and sustained way of sharing information so that the communities have the right information and and also build their capacity. The community leaders put their capacity so that they're taking a leading role in sharing the right information. Right? I think that that's what we need to do. Address the misinformation and the needs that have been going around. I guess let me stop there.
Thank you very much.
Hi, Roger. I just wanted to add on to that. I think we've all had this experience where we've been gripped by fear, especially during this time of COVID-19. But I think there's one thing that we have learned, that during a time of fear, that is when we need to give out as much factual information as possible to try and dismiss the fake news that comes about. Additionally, what's important is building trust with communities as well. And once trust is built with communities as we advance into COVID research, I think it becomes easier for them to as well dispel the fake news that they receive.
Thank you. I'm going to move into something slightly different. We were hoping to have someone to talk about vaccine today, and that wasn't possible for a very simple reason. It's that people are very busy with COVID. And it is something that I've experienced in my everyday job, getting people, I normally work on HIV vaccine, getting people to focus on something else than COVID has been quite challenging. We can talk a little bit about vaccine. There is one question on vaccine later, but the question here is, where is the resource, not only financial, but the people working on something else? Someone is asking, don't you fear that there is a competition for fund, efforts, and health system between the established, in inverted comma, pandemic like HIV, TB, and COVID-19? I'm going to address that to Sharon, the competition, whether it's on science, on resource, and on funds.
Well, I think at the moment we're in an extraordinary time in the middle of a pandemic where most people's lives have stopped or paused, including research in these other areas. At least in Australia, well, in fact, we've had a very small number of cases, 7,000 cases, less than 100 deaths, but an incredible response with over 1 million tests done already. You know, everything stopped. There's no choice. You know, so we do need to mobilise and address this problem because the world has stopped because of COVID-19. I think, you know, there's definitely an argument that not an argument, but an imperative that there's other health issues that we have to be mindful of as well. But I do think we're in quite an extraordinary time that if we don't mobilise every effort that we can, and of course not at the expense of other health issues, but if we don't mobilise, we will be stuck. So I think it's an extraordinary time. It's not a matter of one or the other at the moment. This has to be addressed now.
Yeah, Roger, if I could add to that as well, I think actually the opposite to some extent may be, I mean, exactly what Sharon said, but rather than competing with one another, I think they actually have complemented one another because many of the both systems, infrastructure and whatnot, for example, in HIV, are now being repurposed for COVID. So for example, many of the grantees that have HIV grants have now been asked to see what they can do in terms of repurposing some of those funds now that, as Sharon mentioned, some of the HIV work has been delayed to try to address the urgency of this pandemic. And some of the networks and infrastructures for research in HIV likewise have been repurposed to address COVID.
Thank you. I think
yeah,
you're making...
And Roger, maybe just to complement that, the Global Fund has allowed for repurposing of unspent fund of the current grant that is finishing to address the COVID crisis, and they've added on top of that. So they've mobilized about $1 billion as a response to COVID, which is a pretty quick and significant amount right now for those countries. I saw a couple of questions also a little bit along those same lines about the impact of COVID on HIV and TB testing, for instance. And I have to say that today we see a significant impact of COVID, but this is not because of the technology. This is really because of the lockdowns and because people are not going out and are not being tested. I would just put a little bit of a caveat here also, something probably to watch in the future, is that when you look at somebody coming with fever and cough, for instance, usually the testing would go to TB. Right now, probably the testing is going to go to COVID, and TB might be a little bit put on the back burner, which might also have an impact on TB, which might be negative. But frankly, I don't see today that there is a limitation because of the technology. It's more because of a lack of availability of people not being able to go and get tested the way they are usually.
Thank you. Pursuing this a bit further and coming back to lesson learned, how can we ensure that the decision makers and the experts beyond HIV recognize and learn from the progress in the HIV response? Who would like to take this one? Sharon, maybe?
Really, really important, especially some of the issues around, human rights at the center of the response and equity and support, the global response is what's been prioritized in HIV. Some of the big decision makers here around, particularly in the vaccine world, for example, WHO, Gates, Gavi, Cepheid. What I'm seeing at the discussions with those agencies, it's very much informed by the HIV response. What we might not see is in host government decisions. Each country is feeling the pain of COVID-19 and therefore thinking very much about their own country. The US is getting criticised for that, but I can see that in Australia as well, and I'm sure it's happening in other countries. So it's the country. I think the global agencies are approaching it with the principles that we would like to see. I think individual country governments are under different pressures, so to see a lot more cooperation from our governments will really be important. And we should not. Hopefully, they won't all go into sort of me first mode.
Thank you. We are reaching the end of the Q&A, and I've got one final question, which is kind of opening the debate to the future. A lot of innovation is happening. A lot of new things are being designed, new approach in difficult circumstances with lockdown and so on. How much can we expect will go back to HIV and help us developing new drugs, new tools, and especially thinking about vaccine? How much will go back and help us designing this elusive vaccine we have been after for over 40 years? And I would like to give you really 1 minute, less than 30 seconds each, to tell us your key expectation for a return for HIV. And I'll start with Jim. 30 seconds.
Sure. So, no, I think a lot of the methodologies that have been explored in developing an HIV vaccine, which obviously has been a bit of a frustrating enterprise over the past 30 years, can be repurposed for evaluating corona. We've seen that even now, where some of the companies that are developing a vaccine have leveraged their HIV platform to address coronavirus.
Thank you. Philippe.
Yeah, so on our side, I think the real potential impact is that we will see countries and programs buying more systems, instruments to be able to run COVID tests. And when the COVID crisis is going to be over, these instruments are going to be able to run more HIV tests for sure.
Thank you. Mookho. You're on mute.
Okay, is that better? I think just to summarize, back in the 80s, very little was known about the mysterious HIV virus. And fast forward 2020, we have the SARS coronavirus 2, which is now a mystery virus. But from the approaches that we have learned in understanding HIV viral dynamics and human and host responses, this will accelerate a pathway in knowing our mystery SARS-CoV coronavirus. And in terms of vaccine development and vaccine research, we have 30 years of experience in HIV vaccine development. And to date, we don't have a licensed HIV vaccine. However, that research has not been in vain. And I think the pathway, this has paved a pathway for the development of COVID vaccines, as Jim has said, using technologies that have been used in HIV vaccine research. So I would stop there.
Thank you. Maureen, if you heard me, just what do you think, what will be the return for HIV prevention? What do you expect the return will be for HIV prevention from the work done on COVID? I'm afraid we've lost Maureen. So I'm going to move on. Sorry. Thank you very much, Maureen. And I'm going to close by Sharon.
Hello.
You started
Okay.
the final word. What do you expect the return will be for HIV prevention? And as you are interested in cure too.
Well, I'm optimistic actually that all of this investment, urgent innovation, high level of interest to see, an outcome with COVID-19 will lead to positive outcomes, certainly in the HIV prevention arena. Some of the vaccines that we've never been able to license have not yet licensed, but are interesting technologies, particularly RNA vaccines, DNA vaccines, modified protein vaccines, we may see come to market with COVID-19. So that's very exciting that I'm sure the benefits will come back to HIV. The advances that we will need in manufacturing for COVID vaccines will be at a scale unimaginable to make billions of doses in a very short period of time. Again, that level of infrastructure for manufacturing, should we have a vaccine for HIV, will be of some benefit. I expect antivirals with HIV couldn't do that much better. We've done pretty well on antivirals, but you never know. I think the testing issues that Philippe brought up, the monopoly on pathology-based testing that many high-income countries have, may well change and realise the benefit of point-of-care tests. I do think we will see, because all that investment and innovation is so truncated here with COVID-19, I'm sure there will be benefits for HIV. Finally, about cure, I'm not sure about cure, because the challenge that we face with HIV cure is just so, it doesn't exist. There isn't a parallel with COVID-19, but all science and innovation in other areas always pays off. So I'm sure we'll get something out of it on the cure front, although the science is just very, very different.
Thank you very much. We'll now reach the end of this Q&A.
Hi, Roger.
Yeah. Yes.
Yes, so I just wanted, I think because I saw a number of questions and comments coming around us. Just wanted to end on, I think, on the issue of access, to say, I think as we are all thinking about developing all this diagnostics, therapeutic and preventive tool, it's important for us to make sure that when we're thinking about access, let's not leave anyone behind. So I just wanted to end on that note. Sorry, I didn't hear your question, the initial question.
Okay, so good conclusion. Let's not leave anyone behind. We had a lot of questions. Time is short to answer all of them. We'll see if we can deal with them separately. I would like to thank again all those who participated and all great panelists. Today, I'm going to hand over to Lucy for the closing remarks. And thank you all again from the West Coast to Melbourne. It's a long day for all you, I guess.
Thank you, Roger, and thank you very much to the fantastic speakers, Sharon, Jim, Philippe, Maureen, Mookho. It's really been fascinating, and to all the participants for your questions. For more information on today's presentation, please do visit the IAS website on COVID-19 and HIV, and the URL is coming up on your screen shortly. It's iasociety.org/COVID-19 HIV. And do continue to send questions via email to coronaquestions@iasociety.org. Please also note, as mentioned at the beginning of the webinar, that there will be an IAS virtual COVID-19 conference that will be taking place on the 10th and 11th of July, and abstract submissions are now open up until the 25th of May. There's a link there for registrations as well, and you're welcome to register at any time. So please do sign up and continue this discussion and the sharing of important knowledge. I'd also like to thank the Swiss Agency for Development and Cooperation, STC, and Merck Sharp and Dom MSD, our donors, without whom this webinar series would not be possible. You'll be directed just now at the conclusion of the webinar to any short evaluation survey. Please do take a moment to fill that in. Your feedback is really important to us, how we can continue to improve the series and shape future webinars to meet your interests. And stay tuned, the next one in the series will be on the 28th of May, which is going to be the final one at this stage, and will focus on how community-based organisations have been adapting their services during these pandemic times. Best wishes to everyone, and stay healthy and safe. Thanks for joining us today.